Thank you very much. Danke für die Einladung. It's great to see all of you. I'm very excited to tell you today about Junevity. This is a company that I'm very, very near and dear to my heart because it comes from my PhD project at UCSF, and I'll tell you all about it. Our goal, our dream, is to rewind diseases of aging, and we want to do that by targeting transcription factors. So I started my PhD at UCSF with the dream and this very high-risk, high-reward project of finding brand new transcription factors to take your old disease cells and bring them back to a younger, healthier state. And we wanted to do that in a completely not stem cell-related way, so not Yamanaka factor-related way. How do we find these factors? Well, we use what we call the Rewind platform. This is a combination of computational biology and in vitro and in vivo biology, where we can test tons of new transcription factors and find the right ones to reverse diseases related to aging. We think now is the perfect time to go after transcription factors. Why is that? Well, we have a much better understanding of transcription factors and their target genes than we did in the past with advances in genomics. siRNA really allow for amazing targeted delivery and repression of transcription factors in ways that small molecules have been struggling to do for decades. And we also, like I mentioned, have novel delivery techniques, and we are going after specific indications where delivery is feasible with siRNA. And so I think the combination of those three things means that now is really the perfect time to go after them. What are our initial indications? Everybody ages, and basically every part of me is going to get older, so which part do I go after first? Well, we have three main indications that I'll tell you about today, where we have liver disease, obesity, and skin aesthetics, with a combination of high unmet therapeutic need and then also the aesthetic space because of the great interest in looking young for many years to come. So let's dive into it some more. I have an awesome team of co-founders and a team of scientists working with me. So like I said, I spun out this project from my PhD work at UCSF. My professor, Hao Li, is head of our scientific advisory board and is just a phenomenal scientist in transcription factors and in cellular aging, so it's a perfect combination. And then Rob Cahill is our COO, and he's an experienced previous founder. He started his own company and got that acquired and then led a 100-person team at a public company. He's fantastic. And then I also have John Hoekman, also phenomenal. He's our CSO. And he took his PhD work all the way to a public company and FDA approval. So he's been through this whole process before and is a fantastic leader in this space. And we have great investors as well, advisors, and a scientific team pushing this forward. And we're located in San Francisco, by the way. All right, so let's dive into our science some more, and then I'll talk you through how we're going to move this science into a platform and our pipeline into the future. All right, when we're young, gene expression is nice and tightly regulated, tissues function quite nicely. As we get older, it's a disaster. Tons of things change. So here we have gene expression map where every dot is a gene, and this is actual data of young versus old mouse livers. Things that go up include things like inflammation, and things that go down include like lipid metabolism, mitochondrial genes. Things just don't work well anymore. And so our hypothesis is that just targeting and fixing one gene is probably not going to be enough to fix this whole mess of gene expression changes that are happening. So that's where our network-based approach with the Rewind platform comes in. So our thinking is that transcription factors, which are these amazing regulators, if we can target the right one there, that could rewind this mess of gene expression back to a healthier state. So we developed this platform to find that central node, here a pink dot on the screen, where it is the center of this complex network of gene changes that could bring it back to a healthier state. And so in our case, we use siRNA to target that specific transcription factor, which, in case you're unfamiliar with it, you can think of it like the opposite of mRNA. mRNA tells your cells to make more of it. siRNA tells your cells to make less of it. And then once again, this is actual data of our old mice treated targeting one of these transcription factors. Thousands of genes go back to a happy homeostasis, and that led to better liver function as well. All right. So like I said, we don't think that just targeting one gene is going to be enough, and we also want to take lots of different hallmarks of aging and bring them back to a healthier state. So here we have data looking at how different things actually get better when we do our treatment, and with a variety of different transcription factors. We see increases in mitochondria, increases in proteostasis. Those are things that go down with aging. We also see less of the bad things like inflammation and senescence. Something else that's important, like I mentioned, is that we don't want it to be stem cell related or cancer related. We want you to stay that same happy cell type that you were, just a younger version of yourself. And so on that heat map, you'll see that we have a comparison of what happens when we target our factors versus Yamanaka factors or skin cancer, and they look nothing like that, right? It's a very orthogonal pathway. So how do I find these genes in the first place, and then how do I move them ahead into therapeutic development? So we have about 2,000 transcription factors to choose from in our body. They're my favorite type of gene because they're so powerful and they regulate so many different things. So first what we do is we look at large-scale gene expression, old versus young, disease versus healthy, and look at RNA-seq, for example, to say what is likely the upstream target, the upstream transcription factor, regulating the differences between old versus young. And then would perturbing that transcription factor reverse gene expression back to a younger state. And so we do the gene expression analysis, the prediction, and then we narrow it down to our top targets to test in vitro and in vivo validation to then move that into development candidates. Now, my PhD work was focused on skin fibroblasts because they're a great model for senescence and aging. And so we have a skin aesthetics program that's into the validation stage, but that took about 5 to 6 years of work, kind of slow. Now that we've developed the Rewind platform, we went from idea all the way to development candidate for our obesity and metabolism program in 5 months. And we have a collaboration with Novo Nordisk on this as well. And so what's exciting to us is that this platform can be applied to any aging-related disease. And so we have other indications as well that you'll see that we really want to dive into in the future. But as we mentioned, I founded this 10 months ago. So we can't do every indication right away, but we have lots of things we want to dive into. All right, so just to go over these things again, what are the 3 key things that we think are making it possible now for transcription factor targeting to be the right time? We have a much better understanding of transcription factors and their targets. We can understand these gene regulatory networks much better than we used to. My professor, Hao Li, is one of the world experts in transcription factors, and so is the perfect place to learn about this and develop the Rewind platform. siRNA are amazing. There's like 5 FDA approvals for them already. You can redose them many times. For liver delivery, for example, all you do is attach a GalNAc, which is like a peptide sugar, to the siRNA itself. No lipid bubble required. Inject it into the body, and it goes directly to the liver and only to the liver, which is awesome. But other delivery ideas are also like, let's see, skin, for example. You can do intradermal injections, and it can stay localized and then not go elsewhere. And then for adipose, for example, you can do subcutaneous injections. So some of the initial indications we're going after are ones where delivery is relatively straightforward because, as we've heard today, delivery is really hard. Alrighty. Now, let's dive into our data that we have for the indications that I was telling you about. The first study that we did is where we identified a new transcription factor that we call here RW6. So RW is for rewind, and then it's a different transcription factor, are different numbers. And here our goal was to see if we repress this transcription factor, can we rejuvenate the liver and bring it back to a healthier state? And so the model here is we have a healthy mice, obese mice. So DIO is diet-induced obesity, very overweight mice, been eating French fries their whole life. And we have overweight mice that got treated with our siRNA. And what you'll see is that after just a month-long treatment, they have a significant decrease in collagen, which is an indication that there's less fibrosis. There's a decrease in phosphorylated IRF3, which is also a sign of less inflammation. We have significantly decreased creatinine levels, which is a sign of healthier kidney function, and improved insulin sensitivity. And then when we dove into our histology, we see huge differences in the lipid buildup of these mice. So this is the fat tissue, where red staining is fat. And you'll see the healthy control looks fantastic. The diet-induced obesity looks like there was a murder, blood everywhere, right? Fat everywhere in this case. And then in the diet-induced obesity treated with our siRNA, you see a huge decrease, about 30% decrease in that fat buildup in just 1 month. And this was with a not optimized siRNA, not optimized delivery, just our first study. We're very excited to see these results. All right, next up, the obesity program. Switching gears a little bit here. So, obesity is a huge problem in the world, and in many ways, obesity is kind of like an accelerated model of aging. A lot of the things that go wrong in tissue that is obese looks very similar to old tissue or senescent tissue. There's more inflammation, more fibrosis, metabolism doesn't work as well anymore. And so our hypothesis is, what if we could take the fat cells, for example, or the liver cells, and instead of like destroying them or something, in our case, what if we just want to make them healthier again and metabolize better like a younger, healthier version of themselves? And so that's exactly what we set out to do. And with this hypothesis and with our Rewind platform, we were actually able to get a collaboration with Novo Nordisk on this, which we're very excited about, and because their team of scientists is really helpful in getting us going on these models and the new assays to do. And something that their CSO said in February really struck a chord with us. And so in February, their CSO said that by blocking, editing, or fine-tuning the output of genes, Novo hopes to develop genetic medicine therapies that trick our bodies into burning more fat. And that's exactly what we're setting out to do. So with our platform, we came up with 5 lead targets, RW6 being the top one, and then 4 backups, to look at if we can rejuvenate metabolism. So what's interesting is that, so we use our Rewind platform, which is this computational, right, plus in vitro approach. And then once we identify these targets, we look into the literature and see, has anyone done something with this before? And so this one, from an old paper, we actually found someone did investigate it a little bit. And interestingly, they saw that in a full body knockout, first off, it was safe, which is fantastic. I love having a safe full body knockout of a transcription factor. And next, what's really important is that they ate a ton of really high-fat food, and they didn't gain any weight. You see that plateauing black line? That means they did not gain any more weight. They stayed a happy 30 grams. And then this paper dove into the mechanism for that, and it was because they actually just had a higher metabolism. They burned more calories, which is exactly what our computational tool predicted would happen. So let's dive into our own data on this. So 2 of our targets, RW14 and RW6. Here what we're looking at is qPCR data of a collection of mitochondrial genes and metabolism-related genes and how they change. So the light blue bar is the transcription factor that we are knocking down with siRNA, and the darker blue bar is this collection of metabolism and mitochondrial genes. What you'll see is when you knock down the transcription factor, there's a significant increase in almost every one of these metabolism genes. And these genes go down in obesity and go down when you're old. So increasing them is a good thing. And we're very happy to see that just by targeting either of these transcription factors, we see these significant changes. The next question is always, does it lead to something functional? Why, yes, it does. So here we have this really cool assay called the seahorse assay. So the seahorse assay is this great metabolism assay, which looks at oxygen consumption and pH changes in the media to figure out how much are these cells actually metabolizing, how active are they. And Novo was telling us that if we saw a 10% increase in basal respiration, they would be thrilled, and that would be a huge change. We saw 16 and 23% increase in basal respiration, so we're very excited about that. So just to talk through this briefly, what happens here is that you have your cells growing in a nice little dish, and then they get measured for like how much oxygen are they using, and then you add different drugs at different time points to say like, oh, this is inhibiting mitochondria. Okay, how much is ATP production? Okay, this is like uncoupling everything, and then you get maximal respiration. So it's a really interesting assay where you can kind of dive into the mechanism some more. And so we're very encouraged to see these significant increases in basal respiration in these cells with our targets. All right, last but not least, our skin aesthetics program. So like I mentioned, skin aesthetics was the first thing that we started with at Junevity because it was what I worked on during my PhD, right, with the skin fibroblast data. And so what we saw and what we're excited about is that we were able to go from cells in a dish all the way to a mouse. And recently I was invited to Monaco by the owners of Botox to present on this as well, which was quite fun. So let's dive into it a little bit. First, what we see is that when we target our transcription factor, we go from this mess of gene expression that we talked about earlier back to a much tighter system. Many thousands of genes go back to a healthier spot. And that's just by targeting that one transcription factor. Next, what we then tested are these cool ex vivo tummy tuck surgery samples. So these are literally, people have tummy tuck surgeries, there's extra skin. Instead of throwing it away, they send it to me. And what we then do with it is that we inject our siRNA into it using intradermal injection, and then we look at changes in collagen. So collagen 1A and 3A are the most relevant for wrinkles and aging, and those go down as we get older. They go up when we target our transcription factor. We then did a small study as well in mice to see, do we also see increases there? And we do see a trend towards more collagen production when we use our siRNA targeting our transcription factor there as well. And so what are the next steps here is that we want to do some more sequence optimization and delivery optimization, and also do some young versus old proof of concept as well, because this was just in one age group. All right, so now I've told you about our liver program, our obesity program, and our skin program. So let's do a recap of Junevity. All righty. Why are we different? We think transcription factors are the key. They are super powerful molecules. They can target so many different things, and we think because aging is this super complex thing that we've been hearing about all day, right? Tons of things are going wrong. We need to have a large-scale approach to take those cells from that old disease state and bring it back to a young state. We're also focused on down-regulating transcription factors. Generally, down-regulation, taking something away, is safer and has fewer off-target effects. For example, with transcription factors, if you overexpress it, they might bind to some random stuff that they wouldn't normally bind to. If you repress them, however, you have much fewer off-target effects. Also, we want to treat obesity by restoring healthy metabolism, bringing those cells back to a healthier state so that they can burn those fats and they can have like a healthier way of going about their metabolism. One way that we're seeing ourselves in this market, the one example is, for example, GLP-1s are amazing. They are a fantastic way to lose weight quickly, but they lead to a lot of muscle loss, like 40% of the weight can be muscle loss, for example. And the second you stop taking GLP-1s, you gain the weight right back because the whole mechanism of action is just that you feel full. Your body doesn't like learn to be healthier along the way. So one way that we could be positioned here is you take GLP-1s, you lose weight, and then you use the Junevity product for healthy maintenance at that lower rate. All right. So what are the goals through 2025? Where are we at? So I graduated UCSF in June of last year, and we closed our VC initial funding the week that I graduated. And then since then, we've done what I showed you so far. And then what are our goals is that we want to move the metabolism and obesity candidate through or up to IND. We want to expand our pharma collaborations, right? We have 2 that I've discussed so far with you and continue to work on those. And we also want to generate new targets. This Rewind platform is applicable to any kind of aging disease, and we really want to make a big impact here. And so we think that we can dive into more indications down the line, because our dream is really to unleash a brand new era of rejuvenation medicines, and we think doing that with transcription factors is the perfect way to go. So thank you very much for your attention.